Hello, this is Dennis Eckmeyer, and I'm the host of Science for Societal Progress. During this season, once every 4 weeks, I pick one of the 13 most popular episodes from the first 2 years and post the original interview. These extended editions contain a couple of parts that didn't make it into the final cut and give an insight into the underlying conversation. Supporters on Patreon have immediate access to these versions, by the way. If you are one of them, thank you very much. If not, think about it. www.patreon.com/sci4progress. This time I present to you episode number 11, in which I spoke with Hélène Pidon about genetically modified crops and the European Union.
I'm Hélène Pidon. I'm a French postdoc in a German institute in crop science. I'm working on virus resistance of barley. So I'm trying to resistant genes in the wild plants in order to have plants naturally resisting the disease without pesticides or whatever, but with genes.
Let's go all the way back to the beginnings, let's say. The really original breeding for better traits, how does that work and what does it to the genes?
We are now at the beginning of agriculture. Basically, at first, it was just taking the plant that was the best looking and just sowing the seeds for the next generation. So it was kind of empirical at the beginning, and most of the plants have evolved that way, driven by humans choosing the plant they want for the next generation. And the plants that we are now eating are not at all the plants that were there at the beginning, that were there before humans. And they will not survive in the wild because they are not adapted. They are adapted to agriculture and not to nature. So basically, at first, it was really like, oh, this plant seems better. I will sow this one for next year and eat the rest. And then with the Green Revolution, beginning of, I think we can even start around the 18th century, even before the Green Revolution.
Just before the Green Revolution, what happens to genes if you do this artificial selection?
Basically, when you know about natural selection, the survival of the fittest, let's say, the plant that is the best adapted to its environment will have offspring that will better survive. And so their gene will continue in the generation. And with agriculture, it's not driven by adaptation to the environment. It's driven by the adaptation of what human wants. So basically, even by just choosing the parents that were the best for us, we were already applying selection on genes. We were already selecting traits, selecting even genes and mutations that were naturally occurring. And then these mutations were the one that survive now in the plants. That they were not there at the beginning. They appear and just in one plant that will not have offspring and disappear because it was not adapted. But we made this plant adapted and we made this gene, this mutation survive to the next generation and now be all over our fields because, as I said, the plants that we have now were not even there, were species that didn't exist before humans.
So, but what happens to the genes? Is that just one gene that changes when you do this selection process?
It's everywhere in the genome. It's not even quantifiable. It's like even when you look at wheat has in fact 3 genomes. That means that in fact at some point the genetic material was doubled and then tripled. Like there is for one, like humans are diploid and wheat is hexaploid. That means that we have for one chromosome there is a pair and for them there is 3 pairs. So this was also something that was driven by humans because with these 3 pairs of chromosomes, so these 3 copies of gene everywhere, selection can be much faster and we have stuff that are much better with better yield because everything is related to yield, how many plants you can retrieve for your consumption.
But we don't really have control over what we're doing, right?
Yeah, definitely. It just, oh, this one looks good, I will take this one. And not even knowing what's happening below that. just selecting on what's happening and not even knowing what's happening in the genome, what was the mutation, and not questioning ourselves, is this mutation really good or is it affecting the natural environment? Because just by making agriculture, we change completely the environment. And it's already completely synthetic compared to before agriculture.
And that was up to we're just talking about the time up to the 1800s, right?
Here, I think we are still in antiquity or even before.
Oh, so you were talking about the Green Revolution. What is that?
Around the 20th century, there was a big change in the way agriculture was done. So we moved from farmers doing all the work of selecting their crops and trying to get the best yield possible with putting cow poop on their crops and stuff like that, to something much more industrialized, because there were less and less farmers and more fields and a big need of food. And so more and more things that were previously done by the farmers were moved to companies, because there was something that farmers were asking for. They were asking for expertise outside, giving this work to someone else. So the big change was adding pesticides and to the crops that allow to have better yield, but also having companies that select plants, that make the plant breeding. And at this point, science enter into breeding, not just, oh, this one is looking good, so it will be for next year. But, oh, this one is looking good, and if I cross it with this one, it's looking also good. And there we are still at something very phenotypic, very based on the traits and what we can see of the plants. If I'm crossing those two, I have a better yield, I have a better resistance, so I will choose this one, but still not really knowing what's happening on the gene part. And there was also, we know that DNA exists, and we know that mutation are all the things that can create new traits, new resistance, better yield. How can we increase the number of genes that are interesting for us, the number of mutations interesting for us? And so at this point, we have some stuff that are pretty random, like putting a nuclear source in the middle of a field, just waiting for mutation to appear in plants. And yeah, some will be looking good, and so we will take them.
Wait, say that again. They were putting radioactive material into the field,
yeah,
to see what happens?
A radioactive source in the middle of a field. And I'm talking about Europe, and not so long ago.
So when was that? That was 1900 and?
I think 1970s. It was still ongoing, even maybe later.
1970s. That was already the time where people started to become environmentally conscious, right?
Yeah, but I mean, at some point, we are just changing the DNA, but we were not knowing at all what we were doing. And there is also techniques that enter, so chemical mutagens too, like EMS, so it's one of the most used chemical mutagen. Basically, you take a plant and you put it directly it's a liquid, basically. You put it into the liquid, and then there will be a lot of mutation in the seeds at the next generation, and you will select from that. Because you make new mutation, you induce a lot of mutation, and then some traits may be better afterwards, like you can have gained some resistance, you can have gained a bit of more yield, or a bit of a tolerance to salinity, or I don't know which trait. But when you put it into the liquid, you don't know what you will get. And you don't even know what's happening in the genome.
Let me sum this up for now. So we have the breeding process where we select plants that also have mutations. That's where the new traits come from that we select for, right? So we see a new plant and it has a nice new trait and we select for that.
It's also taking plants from outside, like more wild plants, and then seeing that they have something interesting and crossing them with the plant that you are cultivating. The problem is the more we go in the selection process, the more the wild plant is having a lot of things you don't want in a field. And getting rid of that is very difficult because when you make a cross, you have 50-50, and then you can recross, and then you have 25-75% of, so still 25% of wild. And while you are trying to getting rid of all the negative traits, you can also lose the positive one, and it takes a long time.
So trying to correct when you do breeding and there are unwanted traits that got into your plant somehow, you want to get rid of it. So you take wild original plants and then cross those, and then you try to start over with a selection process until you have all the traits that you wanted.
Yeah, that's it. At the beginning, it was just still based on traits. So you have a plant which is wild and has a lot of things you don't want. Usually it's very low yield, it's very bad looking, but there's something like a resistance to a disease or better resistance to drought, for example. And so then this plant, you will try to cross it with your elite material, the one which is already in the field. And at this moment, you have something which is bad looking, and it's still bad looking. It's a bit less bad looking than the previous one, but it's bad looking. And you just retrieve the one that the offspring that are still resistant, for example, the one that have the trait that you want, and then cross it again with some elite material. And so each time that you cross it with an elite material, you get rid of a bit of the wild part that you don't want, and try to select each time for the one that are the most elite, but still with a trait that was interesting you in the wild in the beginning. And so each time you have a generation, and a generation is a year. So you need at least 10 years to make a new variety. And breeders are doing it less and less, because even if you manage to get rid of a lot of things, you are still like 10 years back in the past, because you have still some undesirable traits that come with your genes. So it's a very long process to just keep just what you need, which can be just one mutation or just one gene.
So you have the original breeding process, and you get all these traits, and you select for them, and then you need to correct something that didn't go well, or you want a new trait in there by crossing another wild plant in there. And then this takes a long time. And then the next step basically was to increase the rate of mutation, to use these agents like radioactivity, X-rays, I think, and this liquids that cause mutations, right? That make changes in the genome. So they quicken that, but you still have to do this whole selecting for the right traits and trying to correct when the trait that you want comes together with the trait that you didn't want, right?
Yeah. So basically, just to explain a bit why mutagenesis occurs, it's because it's so difficult to take a trait from a wild plant. It's much more easy to create it by mutation, finally. Even if mutation is random, it's still kind of easier because you make this mutation appear directly into your elite material. And it's not coming with non-wanted traits. It's coming directly. But you are playing a bit with the dice, like, will it appear or not? But the idea was always to try to go over this fact that when you cross with a wild, you have a lot of things that are coming that you don't want. And also the fact that more and more, as I was saying, the wild plants don't really exist anymore, or it's another species, and sometimes it's very difficult just to make the cross. Just to make the cross, then it's sterile or stuff like that. And so you need to save the embryo by some also techniques that are in vitro culture. And so basically that's why mutagenesis is so used. It's because finally the trait you want will appear in the plant, in the background of the plant that you want. But it's by chance. So it can also not occur. Or you can just miss the one plant that was the one you want.
And now we go to the next step. We have these plants that are where we can take the plant that we are using anyways, and then we can mutate it and select for traits that we want. But now we're doing something different. What is that?
Now, with the knowledge about the genome, with new techniques, we can directly go and see what gene is responsible for what trait. So It can be used in several ways. Either it's easier to follow from generation to generation if your trait is still there, but also you can directly put the mutation or the gene you want in the plant, knowing exactly what you're doing compared to doing it completely random, hoping that it will occur, not really knowing what's happening. It's also allowing you, so for example, I'm working right now in a wide variety of barley, and we are looking for resistance to this virus, and this resistance exists in this wide variety. But breeders have difficulties to put these genes into the cultivated species just by crossing, because crossing is so hard. So what they can do now is, when we have the gene, when we know the mutation that occurred, that we can reproduce with gene editing or transformation of the plant, the gene or just the mutation that we want precisely, just where we want. And without cross, we can have this gene, which is already existing in the natural, in the wild, into our crop. And we can also imagine having potato gene into wheat. It's also something that already exists, but in another species that we will never be able to cross. But just like that, increasing our possibilities to have. So I'm working in gene disease, in disease resistance. So and it's something for which breeding is quite looking a lot also. So basically, we can introduce this kind of disease resistance. And if we have disease resistance already at the genetic level in the plant, that means that the plant is able to defend itself against the disease, just because it's incompatible with the disease. For example, it's kind of a vaccine, let's say. Even if vaccine is also quite controversial, so maybe it's not the right term.
In the wrong circles, yeah.
Yeah. So yeah, the plant will be able to resist by itself without applying any pesticide. So that's basically the idea, to be able to have the plant defend itself, to have the plant that is the most adapted to higher temperature, more drought, being able to grow even in drought, and to have some yield, at least, maybe not the same as if there was no drought, but at least some. Yeah, this kind of introducing new traits into the plant just by changing a few bases in the DNA, precisely where we know it will be useful.
So what you're saying is that actually the actual change that you're inducing using gene editing methods like CRISPR are actually much smaller than what you would do when you would use the previously used more traditional methods?
Yeah, definitely, because we are just introducing, with CRISPR, we can even change just one base, meaning just one letter. Just to give you an idea, the genome of wheat is 10 power 10 letters. It's huge. We are just changing one letter. Whereas when we are putting mutagenesis, we are applying some mutagenesis, it will be tens, hundreds of mutations occurring in the same time, where we don't know where, basically.
10 to the power of 10?
Yeah, 10 to the power of 10.
That's 10 billion, is that right?
Yeah, something like that. I'm very bad with numbers.
It's a 10 and then 9 zeros, right?
Yeah, 10 zeros. Yeah, 10 and 9.
Total of 10 zeros, yeah. And 9 zeros is a billion.
Yeah.
Okay. So it's 10 billion.
I think it's 7 billion.
So you only change 1 in 10 billion instead of hundreds in 10 billion. Both doesn't seem like a lot, to be honest.
It will really depend on the kind of mutagenesis you can have, but it can be more. But it's a single event. It's like doing it one time. And there's also natural occurring mutation along the process. In wheat, we consider that there is 100 mutation every billion base pair in one generation. And the thing is, if you consider just a 1 hectare field, then you can have 20 billion mutation in just one field. It's more than the total size of the genome. So there is hotspot of mutation naturally occurring, but in a 1 hectare field, there will be already a lot of mutation even if we do nothing. We are just choosing the one we want, putting it here, and we can't check every field for just the mutation we want. It's not possible. Some are not even distinguishable by eyes, or they are there for being harvested, so we don't check everything. But basically, the chance that it's already naturally occurring is quite high.
Okay. So we already have a lot of high mutation rate naturally, and then we use gene editing, which is low, but it's very targeted. So, I mean, you do it to have an effect.
Yeah.
So you put you change the genes that count, that make a difference, and it's targeted. Now I heard that CRISPR. So we're talking today on this podcast mostly because there was the European Union has a new law on CRISPR specifically, which is a gene editing method. How well do we know what happens when we use CRISPR? Because there was this study that everybody's talking about that CRISPR actually is not as precise and specific as we thought it to be in the beginning, but how precise is it really? How many unintended changes do we make, and how does that compare to earlier methods, to the mutagenic methods?
It's still quite controversial, this study. It seemed that really the change that we don't mean to do with CRISPR are called off-target effects. It seems that it's really, really, really low, like maybe sometimes it will happen once in the genome. The problem is when we do what we call largely a genetically modified plant, so it's CRISPR or the older methods of transformation, you always go through an in vitro phase. So this in vitro phase is very stressful for the plant. And
what is that? What is in vitro in this case? What does that mean in this case?
To introduce a mutation in a plant, you usually do it in what we call culls that are indifferentiate tissue of plant that we induce. And then we make it differentiate again, and so the plant reappear. We make these cells that we change into a plant again, let's say. It's not very clear.
So do you work basically with a plant embryo? Is it like the kernel that you're working with?
It's a bit more complicated in a way, more easy in another way. That for animals, a lot of the indifferentiate state of the cell is a very short moment. In plants, this state is easier to keep. And so what we do is like we are creating a sort of big embryo, and this is what we are changing. And then this embryo gives a plant, except that with plants, normally you don't have embryos, so we are talking now about changing a bit the cycle for this generation. And yeah, this is quite stressful for the plants because there's a lot of things that are going on during this phase, and that can induce also some mutation that we don't know where they happen. But it's something much less than what mutagenesis with EMS or something like that will do. But we know that this can induce also mutations. And mostly it seemed that the thing that was thought to be off target of CRISPR were mostly induced by this regeneration.
So it's not the actual CRISPR method, but the thing that you have to do in order to apply the CRISPR method.
Yeah.
That is inducing. And is there a way? So first of all, you say this is rare. This is a rare event compared to.
I must say that here I'm not a total specialist of in vitro culture and things like that, so I may say some stuff that are not 100% true. So just want to warn that we are touching the limit of what I know. But it's quite rare, and mostly what's important also to say is that it's done in other process than transformation. It's done in a lot of things because in the modern process of plant breeding, even without mutation, without transformation, genetic transformation, that can be editing or other kind of GM plants that are already on the market, you go often through this phase of in vitro culture. So, for example, when you cross two plants that are non-fertile normally to save the embryo, you go through this kind of thing. Sometimes when you want to speed up the process because you want your plant to be the most stable possible, so you want them to be the same at both allele of the gene. So gene has two copies normally, so you want the two copies to be similar, so the variety you will have will be stable during the time. Breeders are applying something they call double haploidization, which is basically just copying the genome once, so you have exactly the same thing on the two chromosomes. And this goes also through in vitro culture. So this kind of stuff is not only for what is called normally GM. It's basically everywhere in breeding, and I've been for years.
So this is nothing special for CRISPR or gene editing. It's something that people do in other methods too, that induces these.
Yeah.
Okay. So now we come to the juicy part, I guess. You already talked about genetically modified plants, GMOs, but we just heard that. So usually when we talk about GMO, the anti-GMO people, they mean only gene editing, which is the latest of the methods we've been talking about. Is that correct?
No, I think they also talk about the earlier method that are still applied also and still useful. So basically,
but I feel the fight is against the gene editing and not against all the methods that we've been applying for 40 years.
But not only gene editing, also what I'm calling transformation in general, which is introducing completely a gene into a plant. Which is basically currently in the US, for example, you have plants on the market that have been transformed. And this is not made by gene editing. This is made by another method, which is earlier. CRISPR is really something that appear like really became something possible. And we thought about it for plant breeding like 5 years ago, maybe. Not so long ago. It's something very new. But there was already some transformation. So just first, I want to remind exactly so there is this European court that decided that the gene-edited plants should be treated as other GMOs, older GM. But in their definition of GM, they said that a GM is something that a way to change a plant that may have not occurred naturally. And basically, afterward, they just say, are excluded of this plants obtained by mutagenesis. Because we have enough knowledge about it, because it's been so long that we are using it, that we know that it's not dangerous. Basically, they are saying that. So they are saying, basically, that mutagenesis is a form of genetical modification, which is true, because each time that you are crossing a plant, you are making a genetic modification. But yeah, they are just saying that this one will be excluded because we know there is no harm going from that, because every plant you eat have been mutagenized at some point, even in organic farming. And I think it's basically also the idea there is no, in big crops at least, there is no plants that have not been at some point in their ancestors subject to some kind of mutagenesis, artificial human mutagenesis. So if you forbid the use, basically you can't use the crop that have been produced for like the last 50 or 60 years, and you go back to something which is very low producing and susceptible to diseases and stuff that no one wants. Organic farmers don't want it either, because the variety they have are all like that. And if we suppress the possibility to use mutagenized crops, then we go back 60 years in the past, which is a very big difference for the crops.
Okay, so basically what you're saying is we have three fundamental ways that we are directly attacking the genome right now. And one is more like a gunshot method, where you just induce mutations. One is one where we move a whole gene from one species to another, which is
or in the
you call it transformation, or is it called transgenesis? Is that it?
Yeah. Well,
and the third one is the gene editing, and both the last two are actually do less to the genome than the first one, which is the one where you use mutagenesis.
Yeah,
is that?
Yeah,
but the first one is the one that they want to keep, and
because we can't
more
go
modern
right now.
Come again?
Because we can't get rid of it. It's not possible. We can't go without it. It's a very useful tool, and we don't have any material really usable that doesn't have this kind of stuff in their ancestry, basically. So yeah, to go back into the three things. So we have this thing where mutagenesis, where you don't know what's happening, you're just trusting that at some point maybe something interesting will appear, and you're changing stuff in the genome without knowing what you are doing, which is authorized and everywhere. And yeah. The second stuff is what's currently the GM on the market, not in Europe, but some other place, like the US or basically the rest of the world. So transgenesis, so basically you are taking a gene. So we can take it obviously for another species, and that's what have been done mostly, for example, with the BET genes, which are resistant to insects. But you can also imagine just doing it by taking a gene in the same species or in a wide variety and just putting it there because it's absent in your cultivated plants, and you don't want to cross the wild one with the cultivated because of all the change that you don't want that it will make in your plant. So yeah, you are not forced to take it from bacteria. You can take it from just a sister plant or from another plant which is already there, but not the same. Like, yeah, they were saying, taking something from a potato, putting it in wheat. It's still a plant, it's still around. Yeah, so it's also something possible. And the idea, yeah, and so gene editing is just changing precisely one or two base, so two letters in the DNA, to make a change that you know will occur because you have some other studies, scientific studies that say this gene is doing that, and if you change that, it will do that very precisely. The thing is, sometimes this means change a gene which is already there, so it do something a bit different. But sometimes the gene you want is not already there. And so sometimes you will want to put a gene from another plant into your plant of interest, just because you know that it will bring just the characteristic you want. Yeah, and it's just transferring this gene, adding it to the genome, which is also something that kind of occur in natural evolution of the plants. You are just saying, I want this one there. It may take normally 10,000 years to maybe reach this kind of stuff naturally, but I will do it in 1 year. So this is the three things that are currently on the market, or currently possible. Yeah, so to go back to the transgenesis, there was also some improvement in the method to transfer a complete gene in the genome. From the beginning, where it was with a lot of antibiotic genes that were coming with it, because to select the embryos you manage to transform, you have also to know which one are transformed or not. And so basically you make them grow in an antibiotic substrate, and only the one that resists to this antibiotic are the ones that are transformed and will come. But now it's kind of much more precise. It was also that before we were putting this gene in the genome, but we were not knowing where we put it. So it can occur in the middle of another gene, or in a place where it will not be expressed or too much expressed. So it was completely random. And now there is also some methods to be much more precise when we transfer a gene from a species to another.
So, but what the point I was trying to make was that basically we have one method that generates a lot of changes that we don't know, and we have two methods that are way more precise and produce way fewer of changes where we don't know what's happening. Is that did I understand that correctly?
Yeah.
And we're keeping the one and saying, well, it worked well for us for half a century, so, and we can't get rid of it because then we needed to get rid of all our crops. But we don't want the other two.
Yeah.
So
it's politics.
Yeah, so exactly. So what is happening here? You have a more random method that we keep for traditional reasons. That's fine. But if we say there's a traditional method that has a lot of randomness in it, and say, well, that is safe, then how can we justify saying the others that are much less random are not safe?
I think also the one where we are directly applying what we want, that's more recent, and the general public was more aware of what was going on at this point and was maybe not aware of what was going on before. And they just see what's new, and they don't understand everything. Like everyone doesn't understand everything. It's normal. They become afraid. Politics just want to get re-elected and got afraid also because they're also not scientists and don't understand everything. And so they say, let's be safe and not authorize this thing before we have enough feedback from it. I must say that the first GMs were sold in the US quite a long time ago, and we start to have kind of good feedbacks on it. And the GM technique in itself never has caused any problem. There's also a thing that there's something I want to put the emphasis on. So basically there is two major GMs sold right now. One is resistance to glyphosate, to an herbicide, and the other one is Bt crop. So what's a Bt crop? A Bt crop is a crop which has a gene from a bacteria that is in fact an insecticide.
And the Bt stands for the name of the bacterium, right?
Yeah. The Bt stands for Bacillium thuringiensis, and this bacteria is in fact used in field directly. So in organic farming, you can take a bit of the bacteria and put the bacteria directly in your field. The difference is that we don't want the plant to do the same thing as the bacteria is doing by itself. We can put the bacteria, it's natural in an organic field, but having the gene directly in the plant is not seen as correct. Moreover, people are saying, yeah, but it's plants that are producing an insecticide in continuous, it's dangerous for the environment. But this insecticide is a protein, so it's basically the same thing we are made of. It's amino acids all together. It's something which is very fast degraded in the nature, and that basically is made of the brick of livings. It's not a synthetic chemical in the way we are seeing other pesticides. We have a lot of proteins all over us. When we eat meat, it's to have proteins, to have amino acids for ourselves. So it will grow naturally. It's not something that will remain in the soil or something like that. And moreover, it's a very precise. Like there is in fact not one BT protein, but a lot. And those proteins targeting precisely one or two insects, and not the whole insect community, just one or two insects. So it's a very low range of action compared to what can be synthesis insecticide, for example, or kind of insecticide that can be used that are more natural, which are, I don't know, and can be used in organic crop also.
So there is so that we have this bacterium, and it produces an insecticide, and we can put the same gene into the plant, but if we put it into the plant, that's wrong. And if we leave it in the bacterium and put the bacterium on the field, that's fine.
Yeah.
That's what the anti-GMO people are saying. And you say that these proteins are very specific.
Yeah.
How do we know that? Are there human trials to see if they if they have effects on the human physiology, and do we know that they're not dangerous to bees and other insects that we need? How do we know that?
It's even more difficult right now to release a GM, even in countries where it's authorized, than to release a human drug. There is a lot of tests that are done on insects, on humans, on animals. I think there is a lot of studies done, and none of them have proved any adverse effects to humans of these BT proteins. And those BT proteins are obviously tested around a wide, large set of insects before we can say this one is specific to this insect or this one. So basically, one idea also below the GMs is that it's a kind of a shortcut. Instead of crossing for 10, 15 years, you just have your plant in like 1 or 2 generations, which is almost nothing. But the thing is, right now, even in countries where it's authorized, there is so many tests that you should do that it's taking the same amount of time. So it's not a shortcut. It's not really a shortcut. It's just something that you can't do in another way. But that means that there is like 8 years of work to test the effect that the plant can have on humans, on everything, on the environment. But the thing is, as companies are doing these tests mostly, even if it's checked afterward by some authorities, people are not trusting it because, it's a company that makes the test. But companies have no interest in selling something that is not good because it will appear at some time, and they don't want to sell just for 2, 3, 4 years. They want to sell forever.
So you think the companies have a much more long-term thinking than we usually say, right? So usually we say, oh yeah, they just want to make money fast.
Yeah.
But you say, no, they have a long-term thinking. They want something is making noise.
Probably longer than our politics.
Yeah. That's true. The politicians. So if such a study takes 8 years, that's 2 terms, right? And yeah, in Germany definitely 2 terms. In the US that would be 2 terms. I guess in most countries it's 4 years. A term is 4 years. Okay, so yeah, the companies have a longer.
I would say maybe a bit. They have still some financial interest, and the reason also why what we have right now in the market are those 2 kind of GMs are also because it's so difficult to put a GM on the market now that only companies can do that. Only big companies can do that. And what was particularly interesting for Monsanto and the resistance to glyphosate thing is that glyphosate at this time was their own product. And no one else was able to sell it because it was under patent. So they were selling like a package. The GM was not the thing that was giving them so much money. The thing that allowed them to earn so much money was selling the glyphosate with it. So basically they were selling a package. And that was what was interesting for them. But if it was a bit less difficult, then some public institution or what can also produce some GMs that can be not only for commercial reasons, but also for more practical reasons. Like, does a company have any interest in selling something normally that will stop farmers from using pesticide? If they are the ones selling the pesticide, no. It's smaller actors And I'm saying right now that the public sector and the academia is a smaller actor. They are the ones that are able to release this kind of genes on the market.
The public sector have a higher interest in having genetically modified plants?
What I mean is that the public sector would like also to release some GMs. There's this example of the golden rice for Asia, which is still in the pipe for such a long time, and I think will soon be released at some point, which at the beginning was owned by companies. But companies saw that they had not so much commercial interest in selling this thing, which has, in fact, it's a rice which has a high vitamin A content and can improve nutrition in poor countries where rice is a staple crop. Companies realized that it was so hard to manage to get this really high vitamin A content and not very commercially advantageous. And so they let the public sector do it. And right now, I think they are quite close to release the golden rice in the market. But yeah, why improve nutrition? It's not always the thing that will bring you the most money. So it's basically academia which will be interested in this kind of reasons, which have not a lot of money, but are interested in nutrition, in health, stuff like that. Because you can also improve this kind of traits on the plants.
I guess the biggest interest would be the governments in those countries where malnutrition is a big issue, right? Those would be the countries that want the golden rice. Or what I heard now was that there's, in Kenya, they developed a banana that is resistant to nematodes. I don't know if you heard about that. But only recently they changed the laws there so that they can actually use it.
The politicians in those countries are very interested in that because they are seeing directly the interest. And I think we have kind of in Europe and also the US, we have kind of this spoil thing. We are like, yeah, we don't want that because anyway, even if we don't, we can have enough food. We can have whatever we want. In what we call southern countries, like countries that are still with an active development ongoing, the thing is completely different. There's a balance which is completely below the good. There is a big good thing and an adverse effect which is not known, and there is, in fact, so far no known adverse effects, despite a lot of tests. And so what they see is that they can improve health with that. They can improve also yield and have more food. And so there's a completely different view. But I think as Europeans and Western countries, we should also think about our responsibilities, because when we say we don't want that because we anyway have enough food, we are also saying to these countries, maybe it's bad and we are still trying to sell it to you. We don't care about it. So in those countries, they will say, hey, if Europe don't want that, why are you giving it to us? And well, it's not because we don't need it that we shouldn't push it away. It's just a method which has so far, despite a lot of tests, proven no adverse effect. We can do bad thing with it. Like for me, the resistance to glyphosate is not a good thing. It's something that was done also because some farmers were wanting it because it's easier. But it's not something very interesting that we can do. There is so much thing that we can do with GMs, like improving resistance to pests, and so you don't spray chemicals, improving resistance to drought, improving resistance to anything, having a higher yield with lower chemicals, either natural chemicals or not natural chemicals, or synthetic ones. And so, yeah, basically, with also the global warming coming, the environment is changing really, really fast. And breeding has struggled to cope with that and follow the rhythm of global warming. Global warming is right now coming faster than normal breeding can do. But with GM, we can go faster. And we can manage to get more environment resilience for crops and prevent starvation caused by loss of yield because there was a big drought this year. Like right now in Germany, there was a huge drought, and I think they lost 40%. They have 40% less of wheat than the other years, just because we had no rains from April, something like that, almost.
Oh wow, that's
Uh,
unheard of. I mean,
yeah,
I grew up in Germany, and it was always like, yeah, in summer we get rain like once a week.
yeah.
I was also from a
It was
very
like that
I,
last year, but this year
I'm from a rainy
was.
part in
I'm
Germany
from
too,
south of France, and I was feeling at home.
but
Except
oh
we don't
wow.
grow. Except we don't grow wheat at home because we don't have enough rain. And in fact, it was more rainy in south of France than it was here.
Oh wow. Yeah.
So yeah, it's an abnormal year, but abnormal years are coming to be
More
the
normal.
normal stuff.
Yeah.
And if we have plants that can resist more drought, but also that can resist submersion, because in fact last year was not a normal year either in Germany. It was too wet.
I
So we
see.
had a too dry, too wet, and it will be a bit like that every year. We will not know anymore what will be what will the season look like. So if the plants are able to resist by themselves to this kind of thing, we can be more sure of what we have in the end.
Right.
And and I think it's kind of a bad thing to just suppress the tool because we are afraid of what it's doing, because I think it's kind of also a dogma, because when we were not knowing what we were doing, it was like finally just kind of a natural thing that we make faster. But
Hmm.
now that we know what we do, we are basically, I think people are saying that now we are changing stuff in a way we want, so we are playing God, let's say. Whereas when we just do something random, it's random. Uh,
Okay.
but finally, isn't it better to have control on what we do than just make random stuff saying it's not completely our fault if it's happening? So yeah, I think it's also that when we know what we do, we can finally do bad stuff, let's say. But there is so many good stuff that can be done with this tool, because some plants are already resistant to drought. Just they are not our crops, but they are there. The genes are there. They are already in the outside. If we just put them in the plant that we eat, so we can eat them every year, then why should we stop?
Yeah.
Why shouldn't?
So I have your list of things that you wanted to talk about here. And I think the only thing that we didn't go into detail to, and I don't think we have to, would be what is organic farming. I feel like we kind of made a point that it makes no difference because you said at one point that, I mean, you can explain it. I'm not sure where I would put it in the podcast later or if I would just narrate it to make things shorten. But basically, what you said about this is first, organic farmers are also using crops that were made with mutagenic methods, and the second part was that the goal is to have plants that don't need additional herbicides and additional pesticides.
At first, I think something that not most people don't know is that organic farming is using pesticides. Organic farming is using pesticides that are called natural because they are mineral or bacteria or things that you can find in the nature. You don't synthesize them; they are already there. But that doesn't mean they are good. There is a lot of poisonous stuff in the nature, And basically, the two most used things are copper and sulfur, and they are shown to be very dangerous to the environment, to the soil life, to the insects. But they are used, and every year the European Union is saying, yeah, you can continue using it. Because if we stop using it, there will be no more organic farming. Because at some point you need to defend your crop against pests. So there is stuff put on organic farming, which are not better than stuff that was made by humans. Because on conventional farming, we also put pesticides, but these are molecules that were synthesized, that are coming from human chemistry. But there is stuff that doesn't mean they are more dangerous, basically. So I think that's the first point about organic farming. So there is currently in France something called We Want Poppies, basically saying we want to have all synthetic pesticide forbid. But why should we make a difference between what's supposed to be natural and what's synthetic? Because also the natural ones are produced by the same companies that are producing the synthetic ones, just different factories or even the same factories. Why should we make a difference? Basically, it's the same thing. It just the origin is a bit different, but not so much. And why are the organic farmers using it? Because the idea below organic at first was not to use them, but they are still using it because they can't produce without it. But for me, GM is the best future that can happen to organic farming. Because if the plant can defend itself, you don't need pesticide anymore. But if your plant can't defend itself, then you need to put some pesticide on a field. And if you don't have even the synthetic and efficient one, then you will use the natural one, which are not better for wildlife, but less efficient. So you will need to put even more. And finally, you are putting also the same thing, because if a GM plant is able to make the protein of a bacteria, and you just put directly the bacteria, you are putting a living organism that was not here in the wildlife. So you are changing also the equilibrium. So finally, isn't it better that the plant is synthesizing this protein by itself, whereas you put directly the bacteria? So that's basically the point, that finally, for me, organic farming is not a good way to go ecologic, because it's based on something which is absolutely not scientific, which is that what's coming from nature is better than what we can do ourselves, which is basically not true. And the varieties they are using are the same that are used in conventional farming, except for GMs, which are outside of it. But so these plants were also going through mutagenesis and in vitro cultivation at some point. It's a way that we can do breeding efficiently. And when we say that we want the farmers to do their own breeding again, most farmers doesn't want to do this breeding again. The reason why it was externalized to companies is because it was getting more and more complicated, and companies that were devoted to doing that were more efficient than a farmer, which has also to check on his field and do a lot of stuff and has a lot of fields and doesn't have the genetic instruments to do his own breeding by himself. So that's also why companies appeared.
Thank you for this conversation.
Thank you. And good luck for the final stuff.
Thanks. Good luck to you for your work.
Yeah, thanks.
This was it. You just heard the whole conversation. How does it compare to the usual half-hour cuts? Do you enjoy the conversations more, or do you prefer the podcast to have more density? Let me know on Twitter @SciForProgress or by email info@scienceforprogress.eu. I thank you very much for listening. Have a good day. Bye-bye.